Sickle cell disease is a cruel genetic disorder that produces a defective form of hemoglobin, the protein that red blood cells need to carry oxygen and deliver it to the body. The defective hemoglobin deforms red blood cells into irregular shapes resembling sickles. These cells often block blood flow, causing organ damage and sometimes extreme bouts of pain. For one sickle cell patient, Victoria Gray, the disease has been crippling her entire life. Much of her existence has been marked by trips to the emergency room due to pain. With her life and career ambitions interrupted or put on hold, the sickle-shaped cells have damaged her heart. Moreover, many patients with sickle cell disease don't live beyond middle age. Strokes or heart attacks are a constant threat. Around 100,000 people in the United States live with this disease. It's the most common inherited blood disorder and affects millions worldwide, and it's notoriously difficult to treat. Some patients can receive help from medications, and others sometimes undergo risky bone marrow transplants. But for many, these treatments aren't enough to eliminate the constant burden of the disease. When Victoria Gray explored the possibility of receiving a bone marrow transplant, her doctors instead suggested something different. In fact, something profoundly different to try to treat her disease. She jumped at the chance and became the first patient in the United States with a genetic disease to be treated with a revolutionary gene editing technique using CRISPR. And now, 1 year later, it seems to be working better than any doctor could have imagined. The billions of genetically modified cells that doctors infused into her body appear to be alleviating virtually all the complications of her disorder. So what happened inside Victoria Gray's body that's left her almost completely free of the symptoms of her disease? Is the promise of CRISPR as a medical revolution starting to become realized? To understand how CRISPR is used today, let's go back to the beginning of its story. When CRISPR was first discovered in 1987, researchers didn't even fully understand what they had found. While investigating a particular gene in E. coli, scientists noticed an interesting pattern in the DNA surrounding 5 identical sequences, each composed of 29 bases. These repeating sequences were separated from each other by 32 base blocks of DNA called spacers, each of which had a unique sequence, the scientists wrote. The biological significance of these sequences was not known. During the next decade, these repetitive patterns were found in many different microbes. Eventually, these patterns were named clustered, regularly interspaced, short palindromic repeats, or CRISPR for short. However, scientists still didn't know what they were for, but certain observations about them began to emerge. They were always next to specific genes that produce enzymes, labeled as CRISPR-associated genes or Cas genes. The Cas genes encode enzymes that can cut DNA, but at that time, no one knew why they did so or why they were always next to the CRISPR sequence. Soon, scientists noticed something else about these DNA sequences. The CRISPR spacers, the variable sequences between the repeating sequences, looked a lot like viral DNA. With this revelation, the purpose of the CRISPR sequences started to become clear. These repeating patterns are a weapon that bacteria use against viruses. When a virus infects a bacterium, it injects its genome into the cell. If it's a previously unseen virus, a new spacer is derived from the virus's genome and incorporated into the CRISPR sequence. The spacers are essentially a history of past infections, so the bacterium won't be infected again. The CRISPR sequence is then transcribed, creating short CRISPR RNA molecules from those repeating palindromic sequences. Their palindromic nature means that these sequences in the CRISPR RNA can fold in on themselves, creating a hairpin structure. The CRISPR guide RNA then binds to the matching viral DNA. The Cas enzyme then cuts and destroys the invading viral genome with the hairpin structure, helping the Cas enzyme to cut exactly where it's supposed to. With the genome broken, the virus can no longer replicate itself, and the infection comes to a halt. When scientists realized that these microbes were effectively programming their enzymes to seek and destroy specific DNA sequences, the significance of the discovery began to be evident. This could be used as a tool. In the past, researchers had been able to use restriction enzymes to cut DNA, but they did so indiscriminately. In contrast, the CRISPR-Cas system could make precise cuts, giving rise to a new area of science, what we now know as gene editing. CRISPR technology allows genetic material to be added, removed, or altered at particular locations in the genome. Much like hitting Ctrl F on your keyboard, finding a word in your document, and deleting, changing, or replacing it. To harness the power of CRISPR in the lab, Scientists first design and synthesize short guide RNA molecules that match the sequence of a gene they want to edit, usually about 20 base pairs long. Then, like in the targeting step of the bacterial system, this guide RNA shuttles the Cas enzyme, usually the Cas9 enzyme, to the intended DNA target. The Cas9 enzyme then makes a cut across both strands of the DNA. At this stage, the cell recognizes that the DNA is damaged and tries to repair it. Scientists can use the cell's own DNA repair machinery to add or delete pieces of genetic material or to make changes to the DNA by replacing an existing segment with a customized DNA sequence. And this brings us to Victoria Gray and her treatment for sickle cell disease. While sickle cell disease is indeed a genetic disorder, her treatment didn't involve deleting or changing the genes that led to the disease. Instead, scientists found a way to almost completely alleviate the symptoms. The treatment, called CTX001, addresses the disease by increasing the production of fetal hemoglobin, a type of hemoglobin that's present at birth but then stops being produced. The idea is that restoring the production of fetal hemoglobin can compensate for the defective hemoglobin produced by sickle cell patients. To do this, doctors first obtain stem cells from the patient's bone marrow. Then, they deliver the CRISPR-Cas9 components to the cells, where they create a deletion in the BCL11A gene, which encodes a transcription factor that normally represses fetal hemoglobin synthesis. This breaks the gene and allows the cells to resume producing the protein. Billions of these modified stem cells are then reinfused into the body. They won't replace all the existing stem cells, but if enough of them successfully implant and start producing the needed protein, the treatment will be effective. Doctors hope that, after the treatment, 20% of the hemoglobin in Victoria Gray's body would be fetal hemoglobin, but the results have far exceeded expectations. 1 year after the treatment, approximately 46% of her hemoglobin is now fetal hemoglobin. Additionally, 81% of her bone marrow cells now contain the genetic modification needed to produce the fetal hemoglobin protein, indicating that the edited cells have continued to survive in her body for an extended period. Gray's life before the procedure was a constant cycle of dozens of blood transfusions and hospitalizations every year. And now, after the experimental procedure, those numbers have been brought to zero. She hasn't had any pain episodes, emergency room visits, or need for blood transfusions since the procedure. And while Gray was the first patient to receive this experimental treatment, she was not the only one. Patients with beta thalassemia have also received CTX001 to restore fetal hemoglobin production. Their blood disorder prevents them from producing enough normal hemoglobin. However, gene therapy in several of these patients has allowed them to go for months without needing blood transfusions. Is this a cure for these blood disorders? Not exactly, but gene therapy like this can potentially prevent many, if not all, of the complications of these diseases, and the change in the lives of these patients cannot be overstated. However, the treatment's not without its downsides. Just like in a typical bone marrow transplant, recipients of the CTX001 treatment must undergo chemotherapy to eliminate their own bone marrow and create space for the injection of modified stem cells. And just like chemotherapy for cancer, this comes with grueling side effects: fatigue, nausea, mouth sores, loss of appetite, and hair loss. It's not trivial. Gene therapy like this is not the same as taking a pill or a medication. It's more like receiving an organ transplant. And just like cancer treatment, it won't come cheap. Despite the extraordinarily promising results from these studies, there's many questions that still need to be answered. Will the treatment keep working for months or years to come? Will it help these patients live longer? Is it safe in the long term? Will there be any unintended side effects? Although these questions remain, it's impossible not to feel optimistic when hearing about these extremely positive results. And these blood disorders are just one of the many things that CRISPR's begun to tackle. Scientists have started researching CRISPR to reverse genetic blindness, muscular dystrophy, cystic fibrosis, and even cancer. And while gene therapy treatment is costly, CRISPR technology in general is inexpensive to test. In the past, altering a gene using traditional knockout techniques could cost thousands of dollars and require weeks or months of work. Now it costs hundreds of dollars and takes just a few hours. There's still much research to be done, and scientists are just scratching the surface of what's possible with this technology. We cannot yet know the exact effect that CRISPR will have on our future, but it will be significant. Science has progressed toward a much better understanding of genetics, but there's still so much we don't know. In many ways, our genes are a book that can be read to determine our destinies, leading us to good health or sometimes unfortunate illnesses. However, scientists are becoming increasingly aware that DNA doesn't explain everything, and that our environments and experiences play a larger role in how our genes are expressed than previously understood. If you want to learn more about how environmental factors can determine which genes are turned on and off, you should watch our Genes Under Influence on CuriosityStream. It's a 50-minute in-depth documentary that explores how identical twins with the same DNA can be physically different, how generations of plants can be physically modified without changing the DNA sequence at all, and how epigenetics shapes our biology.